Hello everyone, I am Shurosri Pal. I am a Prime Minister's Research Fellow and we are discussing question number 3 from the paper XLQ of GATE examination 2021. The question states, the stationary phase used in gel filtration chromatography is composed of A. Fludextron, B. Carboxymethyl CM cellulose, C. Diethyl aminoethyl cellulose, and D. Saccharose. For more information on chromatography, you can follow this NPTEL course Genetic Engineering. The YouTube link is also provided here. So, gel filtration chromatography is also known as size exclusion chromatography. Now, gel filtration, which is also known as size exclusion chromatography or molecular sieve chromatography. In this process, separation is based on the differing ability due to differing molecular size of the molecules in the sample to enter the pores of the gel filtration medium. So this resin, which is used to pack the gel filtration column, they are beads. These beads have different pore sizes. Now, the stationary phase in this technique consists of beads of a hydrated sponge-like material that has pores of molecular dimensions and with a narrow range of sizes. So these beads, they have pores inside them, and these pores are also they also have a particular size. So there's a range of sizes of these pores. When an aqueous solution, which contains molecules of various sizes, is passed through this column containing such molecular sieves, so these pores, these are the pores where molecules can enter. So that is why they are called these molecular sieves. Molecules which are larger than the pores, they obviously cannot enter the pore, right? So molecules that are larger than the pore, they move quickly through the column as they do not enter inside the pore, right? So these molecules are called to be passed or eluted in the void volume. So suppose this is mm, a particular pore size of molecules that are coming in this solution. So suppose it is here and it cannot enter the pores. So what will happen to it? It will be passed in between in the void spaces between the beads, in between the void space between the beads. So therefore, this molecule, which is a very large molecule, which cannot enter the pores, will be coming out in the void volume, will be eluted in the void volume. But smaller molecules enter the pores of this gel matrix, and they move slowly through the column. Why they move slowly? Because they can enter inside the pore, and then their uh, their flow rate is lesser, right? So the molecules are eluted in the order of their decreasing molecular size. So the smaller the molecule, it will remain longer in the column. So the larger molecule will come out first. So as we can see, these gray ones, these are the larger molecules. They are coming out fast, right? But these molecules, these are the smaller molecules. The smaller molecules are coming out later, right? So the molecules are eluted in the order of their decreasing molecular size. So the larger ones are eluted fast and the smaller ones are eluted later. The molecular mass of the smallest molecule unable to penetrate the pores of a given gel is said to be the exclusion limit of the gel, right? So the molecule which cannot penetrate the pores of the gel, that is said to be the exclusion limit of the gel. So we have these gel filtration resins where there is a molecular weight cutoff. So suppose this molecular weight cutoff is 200 kilodalton. So therefore, it means that proteins which are whose molecular weight is greater than 200 kilodalton, then these molecules will not be able to enter inside the pores and they will be eluted in the void volume of the column, right? Whereas proteins which are lesser than 200 kilodalton in weight will enter the molecular sieve or will enter the gel filtration resin and they will be eluted in the decreasing order of their molecular size or decreasing order of their molecular weight, right? Now, the dimensions which are important to gel filtration are the diameter of the pores that access the internal volume and the hydrodynamic diameter of the protein molecules. Now, if we consider the protein molecules as globular, because as we assume that protein molecules are mostly globular, so therefore there will be a hydrodynamic diameter of these protein molecules, the spherical protein molecules. So this is defined as the diameter of the spherical volume which is created by a protein as it rapidly tumbles in solution, right? So the diameter of the sphere that that this protein can create. Now, proteins whose hydrodynamic diameter is small relative to the average pore diameter of the beads will access all of the internal volume and are described as being included in the gel matrix. So they will be included in the gel matrix, whose hydrodynamic diameter is small um, relative to the average pore diameter of the beads. So the bead's diameter, if that is higher than the diameter of the protein, then obviously the protein will be coming inside the beads, right? So proteins whose hydrodynamic diameter is comparable to the average pore diameter will access some but not all of the internal volume. So here we are talking about proteins which are larger. So their diameter, hydrodynamic diameter is approximately same as the average pore diameter. So it will not be able to access all of the pores, but it will access some of the internal volumes. And then it will be described as being fractionally excluded. So it will slowly come out of the column. Proteins whose hydrodynamic diameter is large relative to the average pore diameter, so larger than the pore diameter, they will be unable to access the internal volume. So it will not come inside the beads at all, and they will be excluded from the column in the void volume. Now, commonly used resins for these gel filtration chromatography are based on agarose or polyacrylamide beads. It can be dextrose for gravity or low pressure systems, and polymeric resins for medium pressure systems. So, depending on the chromatographic system that we are using, we can use the particular types of gel filtration resins. And the choice of the resin depends on the properties of the components to be separated and other experimental factors. Like what is the hydrodynamic diameter of our protein of interest, or what is the molecular weight of our protein of interest. According to that, we will select the resin. So if our protein is suppose 100 kilodalton of weight, then we will use a molecular sieve or we will use a gel filtration resin whose pore diameters are greater than 100 kilodalton, right? So the pore diameter should be greater than the hydrodynamic diameter of the protein that we want to separate by using a gel filtration chromatography. All right, so coming back to our question, what is the stationary phase that is used in the gel filtration chromatography? The first one is blue dextran. Blue dextran is a molecule that is used for measuring the void volume in the gel filtration chromatography. So this is not the correct answer. B is carboxymethyl cellulose and C is diethyl aminoethyl cellulose. So we know that carboxymethyl cellulose is a negatively charged resin. So it is used in cation exchange because it's a negatively charged resin, and TEAE cellulose is a positively charged resin, so it is used in anion exchange. The blue dextran is used for measuring void volume. And the last option that we have here is our answer, that is saccharose. The stationary phase that can be used in gel filtration chromatography is saccharose, and this stationary phase can have several different molecular weight cutoffs, like we can use saccharose 200, so which means its molecular weight cutoff is 200 kilodalton. We can use saccharose 75, which means its molecular weight cutoff is 75 kilodalton. So this actually varies, and this will vary according to our experiment as well. So that is our answer.